At Nautilus Biotechnology, we are developing a single-molecule proteomics platform capable of fueling cancer research. Much of cancer research in the last few decades has focused on finding driver mutations. These alterations to DNA generally encode mutant proteins that set cells on a path toward malignancy, or give cancer cells more selective advantages at later stages of disease. In this way, such mutations are said to drive cancer progression. Driver mutations can point to the biology underlying cancer and may direct the development of precision medicines that target their encoded proteins. But multi-omics studies consistently show that driver mutations are far from the end of the story. In fact, focusing on driver mutations alone can leave researchers blind to the mechanisms underlying cancer and obscure potential drug targets, biomarkers, and other therapeutic opportunities. Here, we highlight 3 ways researchers can leverage proteomics to expand the scope of cancer research. These include understanding cancer resistance to therapies, detecting changes in the tumor microenvironment, and assessing determinants of response to precision medicines. Understanding cancer therapy resistance. Many cancers develop resistance to frontline therapies. Cancer cells are continuously mutating. Drug treatment may select for mutations that enable resistance, but many other, potentially inconsequential mutations will come along with the resistance mutations. It is rarely clear from the mutations alone which pathways are important for resistance. With proteomics, researchers can measure protein abundance and modification levels to see exactly which biological pathways are active in resistant cells compared to non-resistant cells. Such studies can direct the development of therapies targeting these pathways. Detecting changes in the tumor microenvironment with proteomics. Identifying driver mutations provides researchers with clues about what pathways to target in cancer cells. But cancer cells do not live in isolation. Non-mutated cells may support cancer growth and respond to cancer signaling. Non-mutated cells in the tumor microenvironment may thus have altered biology that can be targeted therapeutically. But this will not be clear from their normal DNA. To understand how these cells aid cancer and how they can be targeted, researchers must look at their proteomes to see what biological pathways are active. Assessing determinants of response to precision medicines. Increasingly, cancer patients are treated with precision medicines designed to leverage knowledge of patient biology to target cancer cells. These precision medicines are effective for many patients, but many others still fail to receive any benefit. To determine why some patients respond and others don't, researchers must determine how cancer cells, normal cells, and the therapeutics differ between responders and non-responders. Here too, mutational analysis of cancer cells will be difficult to interpret and will only partially reveal altered pathways in responders versus non-responders. Proteomics, on the other hand, can show exactly what biological pathways are active in responders and non-responders, regardless of any underlying mutations. These pathways can then be activated or inhibited to change patient responses for the better. We hope it is clear that proteomics holds great potential to improve our understanding of cancer biology and enable researchers and physicians to save many lives. Visit nautilus.bio to discover how the Nautilus Proteome Analysis platform can advance cancer research, and check out our proteomics and cancer ebook to learn more.